Bismillahirrahmanirrahim. Assalamu alaikum. Isn't it surprising that there are hundreds and hundreds of types of diseases and they are caused by five different types of organisms? You would be surprised to know that these five types of organisms, even if they belong to the same species, they are very different from each other. They differ in various aspects, they differ in pathogenesis, they differ in their genetic constitution, and they might cause disease in a different way. When I was doing my PhD, I came across two serotype 3 streptococcus pneumoniae isolates, which were different in terms of pathogenesis. Now imagine they were isolated from the same patient, they belong to the same serotype, they belong to the same species, but yet they had different pathogenic capacity or virulence, as you would say. So, as we say, the world of microbes is full of wonders. I am Dr. Yahya Noori, your instructor for this course, and today we are going to talk about the rest of the second chapter from Warren Levinson, and we are going to compare the five classes of infectious microorganisms. So when you are done with this lecture, you should be able to describe the differences between different classes of microorganisms. So these five classes which we have covered in the last lecture, and you should be able to clinically correlate these differences and see what their importance is in terms of disease. So the five different types of organisms which cause infections are, as you know, viruses, bacteria, protozoa, fungi, and worms. They differ in their cellular structure. So you would know that viruses do not have cells in their body. There is a nuclear material which could be either DNA or RNA, and it is enclosed in a protein coat. Bacteria are prokaryotic organisms. So they have a main region where the nucleus is not present, but the genetic material, which is DNA, is tightly packed and is found in a circular form, which is called nucleoid. While there are certain accessory DNA molecules which are roaming around in the cell, and these are known as plasmids. While protozoa, fungi, and worms are eukaryotic, and their genetic material is present within the nuclear membrane. So, since these organisms they all have different structures, we can have different specific drug targets which can be used to target them. So for example, if we talk about fungal cell wall, it is a cell wall which is made up of a specific type of chemicals. So we can use a drug which can target those chemicals, and that drug will have minimal effects on human beings because we do not have those chemicals in our body. So that is an external source of infection which is coming into our body, and that can be identified by different chemicals we take as medicines. Same goes with bacteria. But dealing with viruses and dealing with protozoans is a bit difficult. Why? Because viruses are obligate parasites, they use our own machinery, but they have certain unique features which can be targeted. Protozoans also come in the same category, they are eukaryotic, and our cells are eukaryotic as well. So you need to have a very specific target so that the drug doesn't cause harm to us. So I've already talked about the genetic material. The viral genetic material, we'll talk more about it in the details in the upcoming lectures. It can be either DNA or RNA. So you might come across certain questions. So for example, I might ask you that what if you have an RNA virus? Search what PCR is. You might have studied in your pre-med classes. that there's a process of amplification of DNA which is known as PCR, which amplifies DNA. So what will you do if there is an RNA virus? So how do you detect an RNA virus through a PCR? Let me tell you that. You use a specific type of PCR where you take out the RNA and then you convert it into a DNA which is known as cDNA, and then you amplify that DNA. So that is called a specific type of PCR which utilizes an enzyme from a virus which is known as reverse transcriptase. So we can call it a reverse transcriptase PCR. So you can compare all these properties from here. These are from your Levinson. Now, I have heard somebody asking, what is this S that comes in the ribosome, right? So 70S, 80S ribosome, what is this S? This is actually sedimentation coefficient, or you can also call it Swedberg unit, which has to do something related to the sedimentation rate and the amount of gravity you are applying to the process of sedimentation. Then you are trying to isolate different cellular organelles according to their size. So when we compare the replication, viruses, they disassemble, then they hijack our cellular machinery, they synthesize their components, and then they assemble again. And this is how they replicate. Bacteria are replicated by binary fission, and they can easily replicate extracellularly, while viruses, they have to be inside the cell because they are obligate parasites. While eukaryotes, they replicate by mitosis. However, in different types of eukaryotes, you will see different variations of cellular division, and the types of their division or their multiplication, you would say, that varies among different organisms. Sometimes one organism will form hundreds of organisms. There's another chart for comparison. You might need to memorize the size of the cells, the diameter. You know about the nucleic acid. We have already talked about it. We've already talked about the nucleus. The ribosomes are absent in viruses. There are different sizes in bacteria, and there's something which would be of interest to you, that when we're talking about mitochondrial ribosomes, they are similar in our mitochondria. The size of the mitochondrial ribosomes is similar to the bacterial ribosomes. So sometimes the protein synthesis targeting drugs which we are using, they actually can cause damage to mitochondrial ribosomes as well. That's it for today. I'll see you inshallah again in the next lecture. Assalamu alaikum.